Welcome to another exciting monthly webinar from ICPS. Today is April 28th, but if you're watching this as a recording, it will be published May 3rd, the day before the second annual World Carnivorous Plant Day. Today we are joined by Hannah and Dalton from Pearl River Exotics. They're going to teach us how to propagate Nepenthes. And I also wanted to bring to your attention that we have 5 animated videos coming out, part of our beginner series. All of them will be out by World Carnivorous Plant Day, and we hope you are enjoying them. World Carnivorous Plant Day is the first Wednesday of May every year. Last year we had about 20 videos. This year we have 24 videos, which means 1 video every hour. I'm pretty excited about that. If you want to learn more, you can go to carnivoروusplants.org and then click on the About section and then click World Carnivorous Plant Day. And part of World Carnivorous Plant Day is you can purchase merchandise like t-shirts and tank tops and mugs and totes. And you might be wondering, where does all of those proceeds go to? It goes to our Carnivores in the Classroom grant. Last year, we were able to fund 24 classrooms, $150 each from all over the world, so they can add carnivorous plants to their classroom. K through 12 public schools. If you know a teacher, tell them to be on the lookout this August to apply. If you want to help fund our education program, you can go to carnivoروusplants.org/donate. Okay, so May 4th, World Carnivorous Plant Day. A few weeks later, May 17th, we're having a virtual happy hour, which we do every month, but in May, it's going to be different. It's going to be an open house. Usually it's only for ICPS members, but for May, we're inviting everyone, so tell your friends. On May 30th, California Carnivores is going to be doing the monthly webinar. The time and the topic are to be determined. But then 1 week later, we have Matt Soper all the way from England. He's going to do a live walkthrough I think he has more than 1 acre of glass. So he has this huge greenhouse. He's going to teach us how to grow Sarracenia.
All right, so today we're going to be talking about Nepenthes propagation and comparing different methods, talking about advantages and disadvantages of a few different methods, and then we can, in addition to that, we can get into some of the nitty-gritty of how you actually do those methods. So just as an overview, we'll talk about a few natural methods, so things that these plants do in the wild to propagate themselves. So that would be primarily through seeds, and then the natural generation of basals. Then we'll talk about some human-assisted methods, which would be primarily cuttings and micropropagation, which have some relation with each other, more so than what most people may think. So everybody knows about seeds with Nepenthes. So Nepenthes are either male or female. So in order to generate seeds, you have to have a male and a female, which presents some problems with this being kind of a primary method, especially when you start talking about pure species. So advantages of propagating via seeds would be that you get genetic diversity. Typically a pod has somewhere around 50 seeds in a pod, and then maybe 50-ish pods per inflorescence. So the number of seeds that you can get from a single spike is quite a lot. And then another advantage would be that you have the opportunity to produce hybrids in addition to pure species. Obviously, the reason that a lot of what you see produced in cultivation is hybrids is not necessarily because people just want to produce a ton of these random increasing in complexity hybrids, though some people will be selecting for specific traits, trying to get something that's never been seen before. It's mostly just out of the fact that they don't have multiple individuals of male and female of the same species in order to generate pure species seeds. Disadvantages would be that it's very slow to get plants to maturity. That varies highly depending upon what species you're talking about. So some species that are faster growing, you may just be looking at a few years, whereas the species that are slower growing, you could be looking at anywhere from 15 to 20 years, which is obviously not a realistic time frame for most people. We already kind of talked about the complexities and the problems presented by having to have male and female of the same species for pure species seed. It is, though you do get a lot of seeds from an inflorescence, the production is still limited if you think about it kind of, per year sort of thing, which relates to the fact that it's unpredictable in when a plant is going to flower, at what age, how often after that. Sometimes it's once a year, maybe twice a year, maybe much less than that. And then from there, once you get these seeds, germination rates can vary vastly depending on a lot of things. The primary determining factor beyond your conditions that you have is really the age of the seeds. So Nepenthes seeds lose viability very, very quickly. If you can plant, if you can sow them immediately, off the plant that you just harvested the seeds from, that's best case scenario. Beyond about a week old, it starts to drop off really quickly. Beyond about a month or so, you're really starting to approach a near zero germination rate in a lot of cases. So within about a month or so is the maximum lifespan of viability of these seeds. And then once you sow these seeds, it can be anywhere from a month, several months, sometimes longer than that, for them to germinate. And then after they germinate, it's usually several years. So something approximately 3 years for most species or hybrids to a normal sellable size, which I consider to be about 2 to 3.5 inches. And that's a long time compared to some of these other methods. This is a picture of a male inflorescence from a Diabolica cross hamata. So you guys may remember trying to remember what the hybrid name that was given to that cross was. It's slipping my mind at the moment. Then basals happen naturally. So this is where a node, usually under the media level, activates, and you get a new plant growing up around the base, of the pre-existing plant. This happens naturally with no intervention required by us. And it happens in the wild for sure, especially with certain species. Ampullaria would be the one that primarily comes to mind. Oh yeah, it's Smilodon is the Diabolica cross hamata. And then, so disadvantages of this is that it's slow. Usually you have to have a pretty mature plant for it to start producing basals. It's unpredictable as well in the amount of basals that you're going to get, how many you're going to, when you're going to get them, how many per year, that sort of thing. The proclivity of different species or hybrids to produce basal shoots varies widely between those species and hybrids. So you'll see some that it takes a very old plant for it to really get there, and then others like Ampullaria, for example, it's just what they do very aggressively from fairly early on with most plants. And then depending on how you go about propagating via basal, the survival rates of the basals themselves may vary. So what that's getting at is if you get a large vining plant that then produces basals around it, the kind of two approaches to deal with that would be, you could either cut the basals off of the main plant and then root the basals. Preferably they may already have some roots, which makes it a lot easier to root them. It's going to give you a better survival rate. So really, in that case, the longer that you can wait, the larger you can let them get, the better survival rate you're likely to have. And then then the other way to go about it would be to cut the vine up and then leave the basal with all the roots, which is definitely my preferred method, because then, with rooted cuttings, you're always starting from an unrooted piece anyways. So cut the vine up and then let the basal go about its way with that gigantic root system that the original plant had. A lot of people, seems like a lot of people think that gives you kind of a stronger overall plant in the end if you let a basal grow out like that. And I would tend to agree with that, because they're starting with a much more substantial root system. This is just a picture of a Berkei that we have that was getting pretty aggressive with the basals, and that kind of part on the right there is a probably about 12 inches across or so. It would definitely be survivable on its own if we were to just take that off of there. So with cuttings, the advantage is that the material you're going to use is produced naturally. You don't have to do anything. Really, you're talking about the vining portions of the plant. It's you're typically going to get more production doing rooted cuttings of vine pieces than with basals, just because off of a given vine, you're gonna it's when you take it, you're gonna typically get probably 5 to 10 usable pieces. Something like 2 to 3 nodal segments per cutting is definitely advised. It just gives it more energy to use to root. You're likely to have a better survival rate. Techniques specific to this would be to make your cut at something like a 45-degree angle. The idea behind that just being that it increases the surface area of that exposed tissue. And then from there, the question is, do you use some sort of rooting hormone? We use usually the gel type stuff. It's just really convenient. And we've had fairly good results with it, but I can't make a definitive comparison between using a rooting agent like that versus not. It seems like it's probably pretty close either way, but we do typically use a rooting agent with that. And definitely more predictable than basals, because you're not relying on the plant to really do anything special. You're just wanting it to continue growing its vine that you're then going to cut up into pieces. And then you don't really require any specialized equipment or supplies to do that. Disadvantage is it's still limited production, even definitely compared to seeds. And then we'll talk about micropropagation towards the end. And then rooting rates can really vary. So I've definitely gotten bitten by that before, where I traded an established plant for some unrooted cuttings, which then didn't root. A little bit of that counting your chicks before they hatch sort of thing. So yeah, rooting rates can definitely vary. Things that help are keeping them at higher humidity levels and then giving them plenty of light seems to help. A common practice that you see with doing cuttings is people will cut the leaves in half, or something similar to that. And the idea behind that is, I think, primarily based on that you're decreasing the transpiration from the leaves, so decreasing the amount of moisture that cutting is losing, but with the theory that that's going to increase its survival chances, as well as by decreasing the amount of light that the plant is taking in, that maybe that additionally encourages it to then produce more of its own leaves, encouraging it to start growing and activate those axial nodes. For us, my approach has been primarily to leave the leaves whole, with the caveat that if you're going to do that, it needs to be at very high humidity levels. Additionally, the other question would be, should you cut the pitchers off when you do this? And the best approach that I've found is, in the context of, you've got a setup where it's got plenty of humidity and you've got plenty of light, to leave pitchers on there and actually, put some, say, like a diluted Maxi solution or Osmocote pellets, something like that, in the pitchers, with the idea behind that being that you're going to allow the plant to pull nutrients from those pitchers. And that seems to have good outcomes when we've tried it. So overall, it's still a fairly slow method to use compared to some other options, but it is something that just about everybody could do given their equipment and setups. One downside is that if you're propagating from vines, it can result in ending up with plants which only produce upper pitchers because it's just a continuation of the vine basically. But one thing that you can do to help with that is whenever you put the unrooted cuttings in the media that they're going to grow in, is you set it deep enough to where you have at least one node below the level of the media, so that then it has the option later to activate that node and produce a basal that's going to grow as a rosette like a typical Nepenthes. Different methods for rooting cuttings. A classic one is definitely just to put them in a glass of water, or just ultimately in water, and that definitely works. We are thinking on using water though, is just that it sort of sets you up for then having to acclimate that plant to growing in normal media later on, whereas they seem to root just as well in the media that they'll grow in later on. So you kind of save yourself a step by just rooting them directly in what you're planning to grow them in. So that's our approach. And then there are some alternative things that we've messed around with a little bit. So what's shown in the picture there is kind of a mist-based rooting machine setup. So inside of there, it has a set of misting spraying heads that are spraying up onto the stems, the bottoms of the unrooted cuttings. And then there's that fan there, which is creating a constant air exchange. So it's not getting stagnant. So it's a really cool balance between a few different things that they're getting water, they're getting also air exposure, but it's near 100% humidity. So you're reducing your risk of the cuttings themselves dehydrating. It's kind of a weird experience, but we tried this, and with a rooting hormone in the water of this, and the Nepenthes didn't really root. The cuttings didn't really root. And so then one day I kind of just out of frustration, I just went and dumped that water with the rooting hormone out and just put plain water, distilled or RO water in there. And then they rooted. So in this case, they seem to prefer something without the rooting hormones, which was definitely surprising. So now to get into what I sort of my specialty and what I really want to get into today, which is micropropagation. So we're going to start from a very basic level. What is it? It's propagation in a sterile environment. It utilizes many of the same principles as the non-sterile methods that we just covered. And this in particular was something that it took me a long time to realize. I was kind of conceptualizing it as something totally separate, totally different from the way that we normally propagate Nepenthes. But in reality, it's really the same thing, but just sort of on a smaller scale. So what it's not magic in the sense that it is not going to produce, it's not something that you can just flip the switch on and immediately produce an inexhaustible supply of these things. It's something that takes a lot of work, a lot of money and investment in equipment, a lot of practice, skill, reading academic papers from universities. It's not something to be taken lightly. It's a serious project to undertake. It's also not necessarily the utilization of hormones to to bring out abnormal behaviors from these plants. So a lot of people think that, that it's sort of hinged upon the use of certain PGRs, which is plant growth regulators. That's the sort of more acceptable, appropriate term for plant hormones. You don't actually need them. And in a best case scenario, you won't actually use them. And we'll get into that a little bit later. It's also not necessarily meaning that everything is the same clone, of a particular species or cross. Genetic diversity can be maintained just as well or better with micropropagation as it can from any of the other methods. So why would you choose to go the micropropagation route? And it's, with Nepenthes specifically, and if we were to compare that with other species of plants with different traits. So it's because the other methods are slow or unreliable, and that part of that is contingent upon the fact that Nepenthes are slow growing plants. So if you're wanting to propagate, say a different species in a different genus that is something that grows very fast, that propagates itself very easily, then there would never be any reason, to go the micropropagation route and invest tens of thousands of dollars into a lab when you could just go out there with your pair of scissors and make an unlimited number of these plants in a short period of time. The other thing that makes Nepenthes good candidates for micropropagation is their kind of growth habit of the nodal segments, the kind of vining sort of approach that Nepenthes take. And that sets us up well because we'll use the same principles from either the basals, but really more so the doing rooted cuttings in micropropagation. You can just do it on a much sort of smaller scale where you can effectively make what are structurally the same as the rooted cuttings that you would take from your big plants, but you can take it when they are, say, like half a centimeter long or a centimeter long, something like that. Whereas a piece like that would never be survivable out of sterile culture. It is survivable in sterile culture in micropropagation. So the advantages are it allows for large quantity production, which is primarily constrained by how many of these plantlets that you have, because those are going to be the things that you're going to take cuttings from. Whereas, in a greenhouse or something like that, it doesn't make sense to have, say, 500 individuals of the same species. But in a lab, you could have 500 and they could be genetically unique individuals of that species. You could have 500 of them, in a relatively small area in a lab. And so that's what allows you to maintain genetic diversity, because each one of those could, be its own genetically unique individual. They're also inherently disease-free because they're they themselves are perfectly clean. There is nothing else growing in those containers or in those plants. So they're inherently disease-free, I guess pest-free as well. Disadvantages is that it requires a lot of expensive equipment. So to set up a reasonably capable lab that you're going to have good success rates with is something like $10,000. That's several $1,000s for an autoclave. That's several $1,000s for a laminar flow hood, whatever containers you're using for all this stuff, stir plates, scales, all the supplies, things like that. It's really a huge investment, not only of money to get equipment, but also in time, not only to actually do the work, but there's a lot of time put into learning how to do the work. And that, what are you gonna use as your media? How do you mix all this stuff up? How do you work in a sterile environment and not contaminate these cultures? The other disadvantage is that it is notoriously very, very difficult to introduce non-sterile tissue, particularly from Nepenthes, into sterile culture. So that's like, well, say you've got this really amazing plant in your greenhouse, or in your collection, and you would like to get that specific clone into sterile culture in order to propagate an unlimited supply of it. It is very, very difficult to sterilize Nepenthes tissue, believed to be primarily due to the fact that they're such slow growing plants that these bacterial and fungal contaminants have tons of time to colonize the vascular tissue of Nepenthes. Whereas if you have something that is, say, very, very fast growing, then a lot of times you can just take like the freshest tip off of something like that, and a lot of contaminants will not have had time to colonize that new tissue from that plant. So, usually what this would look like if you were attempting it is soaks and washes in bleach solutions with either dish soaps or other surfactants such as polysorbate, things like that. Washes in hydrogen peroxide solutions, and then then you put this in a container, and it's likely to either die or turn out to be non-sterile, and you're going to end up with a jar full of fungus in a few weeks. So you'll oftentimes hear some objections to what's typically, colloquially referred to as tissue culture, which I should have addressed this at the beginning. Tissue culture, from a definitions standpoint, is really more specific to animal tissue culture, where you would take something like liver cells or kidney cells or something like that, and you would have a culture of cells of one specific tissue only. So you'd have a culture of, blood cells or heart cells or kidney cells, something like that. Whereas in the plant propagation world, we're not isolating a specific tissue type of cell. We are ultimately going for the whole plant. So that's why terms such as micropropagation, sterile culture, things like that are actually a more appropriate term versus tissue culture. We already kind of covered the fact that not every plant in a batch from sterile culture or micropropagation is the same clone. So if you look at this picture on the right half of the screen of that jar completely full of plants, those are some lowland cross involving ampullaria, stuff like that. Those are all seed grown plants in there. Not a single plant in that jar is a clone. They are all genetically unique. And then from there, those could either be just propagated kind of as an entire batch, where they're all just getting propagated together, or you could actually pull each one of those out individually as a numbered clone and maintain each one of those as its own separate clone indefinitely. So, like I said, you ultimately germinate these seeds in sterile culture, and resulting in what you see on the right side there. Additionally, the other option is you could just plant them out at that point. So you've germinated them in sterile culture. You had a really good germination rate due to the optimal environment in there. They grew really fast, something like, 5 times faster. You're going to, plants decent size well under a year, whereas outside of sterile culture, you're looking at 3 years, right? So you could just plant them out at that point. So they were from micropropagation or sterile culture, but they were ultimately seed grown plants that were potted out. The concern about are plants from micropropagation, weaker in some sense, something like that? The answer is definitely ultimately no. but that's contingent upon that they're properly acclimated after they've been planted out from micropropagation. So when they're in these containers, they're growing at 100% humidity. So they have absolutely no ability to retain moisture and not just immediately dehydrate themselves into death. So what that looks like is slowly weaning them down to more reasonable humidity levels. So using things like vented domes, or on a very small scale, maybe like plastic bags, something like that, to very, very slowly work them down from 100% humidity to whatever conditions they're going to be growing in. And if that is done properly, then they are not discernible in any meaningful way from a plant that was grown in some sort of traditional method, other than the fact that they got there much, much faster. And then about the PGRs, there are some concerns about, if prolonged exposure to certain ones of these hormones that are particularly strong can result in a sort of basically permanent alteration to the growth habits of those plants. So you may see clones coming out of micropropagation that do weird things, like constantly want to divide and grow basals and activate every single node on the plant all the time, and you just get this big mess sort of thing. So preferably these things can be mishandled, and they can be caused to behave in weird ways like that long term, through the kind of misuse or overuse of certain PGRs like that. And but this is very easily addressed with Nepenthes. In most all cases, you really don't need any PGRs at all in order to propagate them in sterile culture. And there's really no like super duper secret or anything like that to it. It's once they're big enough for you to cut the plant into a few pieces, you just cut it into a few pieces and then stick those pieces back in the gel, just like what you would sort of very, very small cuttings that you're going to root from the greenhouse, And so therefore, in that case, using that kind of method, you're never exposing them to any hormones that may cause weird reactions. And then if you know much about kind of micropropagation, a fairly typical way to go about it is through the induction of callus, which is where you'll take a piece of the plant and then expose it to usually a cocktail of 2 or more different PGRs in order to induce that tissue to revert into a kind of mass of undifferentiated cells, almost with a tumor type look to it, something like that, where it's just a mass of undifferentiated cells. And then what you can then do is then later get those cells to redifferentiate into kind of each of their own individual plant. So that's where you can get some really, really crazy numbers of plants. You can have masses, like single masses of thousands of plants in those sorts of cases. But the downside of that is that it is requiring some really aggressive cocktails of hormones in order to induce that sort of behavior. So my personal approach, and the approach of other people who are doing this, fairly well, I think is overall to just really not use any hormones whatsoever in their micropropagation outside of situations that really, really require it. An example of that would be like a species that wants to grow in a really, really like tight, flat rosette that really no matter how long you grow it out in a container, it's never going to give you the sort of vertical length that you need in order to be able to take any cuttings at all with it, to be able to chop it up. And in that case, the gibberellic acid, which is used by, as a means to induce germination in certain plants also has the effect of lengthening the nodes. And so a very, very slight exposure to gibberellic acid can cause those nodal lengths to increase to a point where you can then cut them up and propagate that plant. So in conclusion, we talked about seeds versus basals and cuttings, the relationship between basals and cuttings, where you can kind of just cut up the vine, leave the basals, that sort of thing. And then we got a fairly in-depth look at micropropagation as an endeavor and the comparisons between those different methods. And then the ultimate, I would say, conclusion of all that is that for most people, it's the basals and cuttings that make the most sense, just because it doesn't require multiple plants, it doesn't require any sort of specialized equipment or training or a unreasonable amount of time. And then for those, or financial investment. And then for the seeds, that's a little bit more specialized, because then you're either going to have plant, male and female plants yourself that that flower around the same time, or the trading of pollen and stuff like that through the male is fairly common. So you're going to have to arrange something like that. So that's kind of the next level up. But I'll kind of leave you with this, that if you ever want the option of having a hamada side salad with dinner, micropropagation is going to be the answer.
At the beginning, you were talking about rooting gel. Do you have a preference on your favorite rooting gel?
Oh yeah, do you remember the brand of it?
No, but I could probably look it up.
Yeah, it's the one. It comes in a black
bottle.
bottle. I can't remember the brand. It's super popular though. If you go to any of a hydroponics store, they always have it. I think Hannah's looking it up right now. We'll get that in a second.
Very good.
I think it's Clonex.
Yeah, Clonex, the Clonex gel.
Okay. Can you speak to hermaphroditic flowers?
I haven't seen any of that personally, so I can't really speak to that. I have heard, you will hear kind of that people have had instances of Nepenthes flowering as one at one point and then another one later. I've never, like I said, I've never seen anything like that personally, but.
I sent you a link to the rooting gel if you wanted to put it in there.
Cool.
I'll include it in the show notes.
Cool.
James from Florida says he used Bonatide rooting, that company, for rooting powder with great results. And he gets lots of basils from that brand.
I'd be really interested to see it, if somebody really wanted to dig into this and kind of do a legitimate comparison of, cuttings with and without some sort of rooting agent, to see, because if you've only ever done one thing, the fact that you had great results may mean something.
Yeah.
But it might also not mean something. And so yeah, I like a kind of round robin comparison of some powders and some gels and nothing at all versus water. The problem with that is that being able to make a fair comparison, you would really need an incredible number of cuttings, preferably all from the same plant, right? Because if you're comparing like different species or different crosses, then that's really not, it's not like a legitimate experiment either.
Yep. Have to have a lot of space.
Yeah, and time and and cuttings.
Future idea for YouTube video.
Yeah, that'd be a good one.
Yes.
I'll get to work on it.
So Don wants to know, when doing vine cuttings, is the cut piece going into sphagnum or something else?
Yeah, so that's kind of what I was getting at with, you can just put it straight into what you can just put it into a glass of water. People have been really successful with that, and I think I messed around with it as well and been successful with it. but yeah, it can go into effectively whatever mix you're planning to grow it in, though you don't really want it to probably be something super airy, like straight cocoa chips. They just don't have the moisture retention. You're gonna, they're just gonna dehydrate before they ever root. So either a pure sphagnum, sphagnum perlite, something like that, or more recently we did quite a few with putting them into straight cocoa pith or cocoa peat, that kind of thing. And I was pretty skeptical going into that, but ultimately it ended up working out fairly well. They really just need something that's going to keep them, keep enough moisture around them.
Rochelle from the Philippines wanted to know if you've compared the growth rate from cuttings in different media.
Oh, another really popular one that I didn't mention is rock wool. If you've ever seen that, they sell it at hydroponics places usually, but it's basically like it's a spun. My understanding is it's like a spun molten rock, and it has a good balance between moisture retention and aeration. So the question was the different growth rates of rooting between different medias.
It's been about the same.
Yeah, it's about the same all around, which is why I'm pretty apathetic whenever it comes to whatever you stick them in like that, because it seems to work in just about everything. The only thing I would caution against is something that's super airy, like like a straight perlite, unless you're running some sort of hydroponics like setup where it's getting watered really, really often. Then ultimately they just have to have enough moisture
and humidity.
Something like another idea that I've had that I haven't tried is that would probably work well is like a big kitchen sponge, right? Like anything like that is likely to work and be about the same as anything else.
Yeah. James wants to know, do you like triple wash the cocoa peat, or do you have a specific brand, or how does it work for you?
From what I've seen, the highest quality brand is the Canna. I think it's CANNA brand, but it's still not. I think if you look at their website, they kind of say that they remove all the salts from it, but if you actually soak it and measure it, it's in the 50 ppm realm, which is actually really good, but it's not zero.
Yeah.
So, and I don't have a hard and fast rule about how many times it gets soaked or washed, but my approach is just to actually measure the total dissolved solids, or if you do the conductivity, and just keep soaking it for 24 hours at a time, with water changes until it's below 100. So 100 is my threshold. If it sits in the water for a day and we measure it and it's below 100, then I'm fine with it. Usually that takes about 3.
Yeah. And a lot of other countries are using a lot of cocoa products. Like Devin in Trinidad and Tobago says he's had great success with cocoa peat and propagating.
Yeah.
So we've really, due to I don't know how it is now exactly, because we've just about completely switched to cocoa products, but getting a hold of the New Zealand long fiber sphagnum was so unpredictable and difficult that we just completely switched in any case that we could. And it's also a lot cheaper. It lasts a lot longer in terms of not breaking down. The only downside is that you have to wash it, but that's basically a one-time deal. Once it's washed, it's washed, and that's it.
And I generally do most of our watering, and I very much prefer the cocoa products because you get a much more even distribution of water through the pot. Whereas like with sphagnum moss, you can sometimes end up with a gradient. Like if there's a pot that you miss one time, you can water it and the top will get wet, but the bottom of it will be dry. So you really have to go around and kind of pick up all your pots to see whether they're getting watered thoroughly or not. Whereas with the cocoa products, you don't really have to do that because it's, like I said, you're gonna have a much more even distribution of water through your pot.
Yeah, the other thing would be that everybody's run into this probably, that if you let the moss get dry enough, it becomes hydrophobic, really, and you have a ton of trouble rehydrating it. You don't have any of those troubles with the cocoa products. So I think that I feel like that the cocoa stuff mainly came from the like Southeast Asian growers, it seemed like, and I really think that they got it right,
Yeah.
and they had a lot of success with it. So.
Okay, so you're using the cocoa peat, or do you ever use the cocoa chips or bigger pieces?
So for rooting, it's just about got to
Yeah.
be the cocoa peat
Yeah.
in order to have the moisture retention. But then from there, our current preferred mix is about a 1-to-1-to-1 cocoa peat for light cocoa chips,
Okay.
something like that.
Megan wants to know if you have a plant putting out or putting off basils, is there ever a time where you should discourage the growth of the basil?
There's really, as far as I'm aware, there's really not a whole lot that you can do to discourage a plant from producing basils. If it's, as long as it's not, I guess I've just seen a few particular individuals that would sort of try to basil themselves to death, which outside of that, like, I don't really think that there's a whole lot you can do even in those cases. It's just going to do what it's going to do. And in most cases, it's a nice thing because it's giving you more plants. I will add though, I've noticed with, say, ampullaria in particular, which I think this probably applies beyond ampullaria, is that the light levels will affect their proclivity to produce basils. So in low light, they're going to tend to want to vine, and that's because they're trying to reach for the light. Whereas if they're getting plenty of light, then they are going to be more inclined to produce basils in order to, because if you think about ampullaria kind of growing on a forest floor in a canopy covered area, if it's getting light there, then it's just going to grow there and produce basils. And so I do think that maybe the one way you can kind of control it is either upping or lowering the light.
We definitely have one more question, but could you share the name of the cocoa peat product again, or the
Canna,
brand?
I think it's CANNA.
Okay, CANNA.
Make sure that's right.
Yeah, I think I found it. Okay, so then the last question I think I'm going to ask is, have you seen, because John Endy have you seen people grafting Nepenthes?
We've definitely never tried it, but I think, super cool stuff if you made sort of like a, basically like a multi-species plant. But
Like
I guess I view that as being like, it's more of like an interesting curiosity sort of thing. Like it is really interesting.
At the stores, they call them cocktail trees.
Yeah, you're gonna get something like that. So if you're into that sort of thing, definitely run with it for sure. Super cool stuff.
So my background is in agriculture, so I learned about it, in like a fruit tree aspect, because they do a lot of grafting with fruit trees. So it's definitely something that I would like to experiment with.
Yeah, I guess you're the one.
But we have not.
Yeah, I guess I could see maybe like the way that they do it in say like pecans is they'll take individuals that, or in other fruit trees, maybe is you can maybe take a tree that like develops a root system really quickly, and then you graft a limb on from a mature tree that's flowering, that sort of thing. So I mean, maybe there's the potential to like graft the end of your vining villosa on top of a ventricosa, right? So like you have this thing that's very aggressively producing a giant root system, and maybe you would get something really special out of that.
Which, and another thing that would be cool is if you could take, so say you have a mature flowering villosa that is male, and you end up getting a hold of a female clone of Velosa, then, but say like, it's 10 years younger than your mature male plant, there's the possibility that you could take that younger female Velosa and graft it to the mature male and potentially end up with a plant that would flower sooner than if you had done that. But
Yeah.
all
That's
that's kind of hearsay.
That's like on the very sort of like edge between, like
a lot of things would have to go
between
right.
like reality and science fiction, in my opinion.
John was thinking that maybe people could do a fast-growing lowland and then put a Highlander on top of it,
Yeah.
which is kind of what you guys were talking about.
I feel
I was-
like you might end up with a plant that just doesn't grow well in either then. Like the top hates the warmth and the bottom hates the cool.
Yeah. I know it sounds like science fiction, but all these other branches of agronomy and agriculture are doing it.
I guess I sort of I'm a little bit skeptical about people's willingness to chop up certain ones of these plants like that,
Yeah.
The ones that you would really want to do, it's just they're so valuable. You kind of don't want to go and like cut it in half.
Yes. All right. So in the chat box, I just shared the Canna gardening cocoa stuff. And I definitely want to thank Dalton and Hannah from Pearl River Exotics. You guys were wonderful.
The International Carnivorous Plant Society wants you to be successful with your plants. We welcome growers just getting started all the way through professional scientists. We started an annual World Carnivorous Plant Day to celebrate these spectacular plants. Take a look around our website and you'll find historic documents about carnivorous plants, growing guides, free educational resources, and more. Have questions? Ask. We don't bite, but our plants do.